Major Issues Biology Series. I'm George Bruning from the Pathology Department. And I had the good fortune, more than 3 decades ago, to work in the same laboratory with Roger Beachy, laboratory in Milton at Cornell University. And that was indeed a pleasurable experience. I've not run into many people with at-the-bench lab skills that Roger has. I say has.
Yeah, they don't work.
So, Roger, well, I remember those days because we would each work during the day and do calculations at night, come back the next day with a new experiment design. It was great fun. So, Roger is a graduate of Goshen College in Indiana. Following his graduation from Goshen College, he went to Newfoundland, Canada, where he was a high school teacher for a year before he proceeded to Michigan State University, where he got his PhD with Dr. Ruth. And it was after that that I met him, because he then moved to Milt Zaitlin's lab at Cornell University. And there's a streak in Roger. It's a sort of a humanitarian streak that I very much admire. And it started with that teaching in Newfoundland and has permeated his entire career, his interest in advancing people's interests on an international scale. It's really great. He's also a fantastic carpenter, although he tells me that he's not had a chance to touch the saw blades.
Fingers are intact.
Yeah.
No thumbnails broken.
But we both learned the dictum that you always want to have 3 teeth in the work. And Roger has always had enough teeth in the work to make things work very well. Roger Beachy.
Thanks, George. Thanks very much, George. George really is modest here because he taught me an awful lot during that year when he was on sabbatic at Milt's lab, and what I learned from George was first of all never stop. I mean, I think we did 3 experiments a day, and some days, because they were fast, they were in vitro, and you could do them quickly, and mostly the chemistry that he brought to biology was a real high point for me. And we didn't do a lot of socializing, but we sure worked a lot that year. But again, it was upstate New York, and what can you do when it's cold? And as you know, Ithaca, I think it used to at one time had the highest level of undergraduate suicides in the nation, and we certainly weren't going to be depressed enough to do that. So we just worked instead, and enjoyed it enormously. We really did have a good group from which I think we all went on from. And the fact that George is actually considering changing his degree of emphasis at the university, the R word doesn't come into play here, of course, it says that there are changes that can happen. You can still get your career advanced and get your work done that you want to get done because you love it. And I think that's what drove us when I learned from George when he was on sabbatic leave. So what I thought, as I thought about what kind of talk to give today, there are a couple of things that I could have done, and at the end, because you have Bill Lucas here and other people who work on things like plasmodesma, I'm going to that part of my talk I'm not going to have very much of, but I want to talk about the part that really drove us through and still does with regard to how we use our knowledge for benefit, and I want to talk about what's happening in the world of transgenic resistance to virus infection and maybe what the futures are and what the future is and the challenges. And it'd be a lot of fun if you had questions as I went through that you interrupted me. If I don't make it through the slide set, you're probably better off. But it'll also be more interesting. So please don't hesitate to interrupt. I'm going to talk about whether or not the resistance strategies for virus disease. You can argue whether or not they're important or not to study viruses for the sake of resistance. We are in the U.S., after all, and the challenges are very different here than they are in India or Southeast Asia or Africa. But I want to review a little bit some of the strategies that have come out over the years and ask ourselves if there are new ones or not. And you'll find as I go along that that I'm a protein person and not an RNAi person. But I'll mention it just in passing because you probably would challenge me if I didn't. But I want to talk about strategies that are being used. I'll use TMV as a model, but I won't spend a lot of time on this because a lot of it's been published. Then I want to update you on a project that's relevant, and that is a coat protein mediated resistance strategy in real world. Why would you bother doing it if it's not valuable? And that's what I challenge our faculty and students and postdocs with. If you're going to do something, choose it to be interesting and exciting, but if possible, relevant. I want to talk about another virus, a pararetrovirus, and then I'll finish off by talking about a disease that's common to one of my colleagues, and I've worked on a little bit myself, but mostly it's Claude Fouquet's work. And it's been a long time since the first field trial of a genetically engineered food crop. I mean, this is 1987. And we had resistance described in the literature in 86. And this is the thing that I still puzzle over. What relevance will academicians be in developing new products that come out of the new genetics? Because we aren't relevant now. And you'd make the point that unless we're relevant, you'd ask whether or not the legislator ought to fund us. And if we're not relevant, why not? Why hasn't there been more success? And what steps can be taken to enhance contribution in food production? And this is Rob Fraley when he was younger, and that's my hair when it was younger. The graduate student who did this work was Trish Powell, and that's Trish. So the strategies for resistance have really gone all over the map. There are a lot of ways that people are doing pathogen-derived resistance and non-pathogen-derived. And we know those. We know the RNA-mediated resistance strategies. Some of these will be sequences that come from these genes or others. And then there are the non-pathogen protein-mediated resistance strategies that have been come into play lately. Transcription factors, DNA binding proteins, interferon-like strategies using double-stranded RNA degrading enzymes, translation initiation factors and cofactors, which are known to affect what RNAs are translated in the host. And there are examples where expression of specific translation factors can overcome virus susceptibility to virus infection. So there are a large number of different strategies have been employed, had been developed, tested, and employed at least in laboratory settings. And I'll come back to this later. The numbers of these that have made it to the field have been relatively few. There's a squash variety developed at Seminas, papaya, which is a public sector. There is a new apple variety, a plum variety that's resistant to plum pox virus from the group at West Virginia. Those have all been approved and deregulated and ready for commercialization. The plum are not yet in commercialization because it's quite clear that Europe won't import the plums if they're transgenic. It's an interesting subject because it's also resistant to the European strain of plum pox, and the challenge will be for them to consider how they restart their own plum industry since we now have the resistant germplasm. Will they import it for growing there? So I think it might actually push some buttons at the right time that we hope that it goes in Europe. So the protein-based strategies are what I'll talk about today. I'm going to talk a little bit about the simple one. And I'm going to, what we find out is that coat protein is one of the regulatory molecules in regulating how the virus works. And you take the strategy, if you're a protein chemist, that if it's a positive regulator, that you can create a negative regulator and make it switch it from a positive to a negative type. Then I'm going to talk about a real-world example in tobacco streak. I'll talk about something here on rice tongue grow. The reason I'm talking about novel approaches is because the other strategies didn't work. And then G5, which is a protein from an inovirus, which is a DNA binding protein and has some applications in Gemini viruses. So I mean, if there are lots of viruses in the world of biology, and plant viruses are segments of these. There are plant viruses that play in in different classes, and they're indicated as these colors of green, the single-stranded plus sense RNAs. And this is a different kind of genome. It's got antisense. Here's a set of viruses that have double-stranded RNA as their genome. Others have double-stranded DNA. Some are integrative. Many are not. And they are in different categories. So I'm going to talk about this category as the largest variety of plant viruses, as most of you know. And the example I'm talking about is this guy. It's a very simple virus. I started studying it when I was a graduate student in plant pathology at Michigan State. And to be honest, the reason I chose it was because it only had 3 proteins. At least we thought it only had 3. And I felt sorry for the people who studied fungi and bacteria and figured I was not smart enough to study those. So I thought I'd study a virus instead. Maybe it would be simpler. But I've been working on it for a long time, and a lot of people worked on it before I took over, before I started. So it's got a historical significance. This is a slide from Milton Saitlin's lab. And it really we really worked on how does virus replicate from the standpoint of molecular biology. How does it replicate? From the standpoint of cell biology, we had developed this cycle while I was and published it a number of years ago. And so in the context of a cell, where do all of these processes happen? And if they happen, where is a weak link in this cycle that could be interdicted by some strategy? And we had a number of strategies that we made up. But it's the knowledge of the cell biology that has informed this strategy. This knowledge was used to do coat protein mediated resistance. And once we know this, we try to understand this in the context of this. Now, in the context of the virus itself, we also had a lot of information about structural biology of the virus, and the early data still holds, and that is this resistance reaction is dependent entirely on having large amounts of coat protein. Furthermore, we made mutations that prevented virus assembly, could make a lot of the monomer. When we made only the monomer, we did not get resistance. We had to have the ability of the monomer to assemble into multimers, including trimers and larger, and the ability to co-assemble. So there was kind of a capping reaction that went on in some way that drove a reaction. The early stages of virus infection drove it back towards assembly. And that we showed relatively early on in the process of characterizing resistance. And that fit in nicely with what we knew here. But this guy kept bothering us. And when I was at Scripps, I had the privilege of working with some outstanding cell biologists and microscopists, and then have continued through the collaboration at the Danforth Center with Howard Berg. And I'll touch on this a bit too. But first, a little bit about the structural biology. In a paper they published last year, we made mutations in the coat protein. And that was of course made possible because of the work of Gerald Stubbs and others who helped us with the quaternary structure of tobacco mosaic virus. We knew about the interactions between subunits in this direction and subunits in this direction. So we knew how the thing went together. We could then do point mutations, which changed the ability of the virus, of the protein to assemble or not assemble. So we did the simple experiment, made lots of mutations, put them all in transgenic plants, identified plant lines that had the same amount of coat protein and said, are you resistant or not? Did the same thing with the virus and asked if it assembled, caused systemic spread, and so forth. And just a little data from that work. So what we found is that different mutants have different phenotypes. There are some mutants that don't give resistance. They get, this is a disease susceptibility plot. Plants get sick, as shown by this dot. When they're sick, they have a lot of virus in them. Here are some coat protein mutants. These are transgenic lines that carry these mutants. And they don't get, there's almost no protection there, not significant. Then there are a group of proteins, this group, the wild type coat protein and these mutants, that have this level of protection. They give a delay in symptoms and then get sick, less severe. But then there are other mutant coat proteins where you get immunity. You simply can't get infection, but it's protein mediated, not RNA mediated. It's extremely resilient resistance. And that's this group. Now, you notice that some of these are in proteins that affect this interface, are mutations that affect these two amino acids. These are two repulsive amino acids, as you can see, two acidic amino acids. In the virus, these are held together by calcium, which is bonded between in this large space and holds the virus together. So that calcium binding site was an important part of the reaction. If we change this to a basic amino acid or this one, and we could change the affinities, but still we increase the affinities to the point where we could have very high levels of resistance. Now, we studied a whole number of these guys. What we did was take this information and then make cell lines of the tissue culture cell line BY2, and then studied virus replication in those protoplasts that were transgenic in order to see if we could have resistance at the cellular level. And in this experiment, we overcame resistance by the original stage of resistance that requires the coding and the encoding part by challenging the virus with viral RNA to see if it was resistant at a second level. And in fact, it was. Here we are just simply measuring the amount of virus that goes from inoculation to 36 hours in a single virus cycle. And we found that some of the mutants prevented the production of coat protein. Some prevented, and many of these are in the same grouping, prevented the production of replicase. Some prevented the amount or accumulation of movement protein. The movement protein was one that we've been studying for a number of years and are really interested in how this works, because this is the guy that does two things. It sets up the virus factories, and it's essential for cell-to-cell spread. So we really looked at we applied our knowledge of virus replication on that black life cycle that I showed you earlier to studies of these mutant proteins. Then we took the cell biology approach, and this slide is one to show you what we've done here is identify replicase, movement protein, and coat protein with different antibodies in the cell, in the protoplast, and at 14 or so hours after inoculation, we simply find out where those proteins sit. And you can see that the blue guys, that's the coat protein, and the green guys, the movement protein, and the red guy. This is the replicase. And if you look carefully, you can clearly see that in some cases, the replicase and the movement protein co-localize. These turn out to be factories of virus replication, and we've done studies to follow what those factories look like over time. And so we looked at these protoplasts that are transgenic and said, what do these factories look like in your cell line if you make a transgenic protein, or the wild type coat protein, or no new protein. And lo and behold, they fell into three categories. Those in which it looked like there was no virus, like there was no transgene. Other cases, it looked like the wild type coat protein. In other cases, they were all like the highly resistant lines. And so what this represents just a fluorescent picture. This is a fluorescence microscope view of the accumulation of the movement protein. In this case, it's a live cell image. It's movement protein with the GFP on it. And in early stages, these are found in small puncta. And as these grow, these factories grow in size into large bodies. And in late stages, the movement protein fluorescence is deteriorated as a consequence of proteolysis. That's a proteasome-mediated degradation of the movement protein that we showed some years ago. And in fact, this is a late stage of infection. So early, mid, and late. And so a technician sat at the microscope for like 6 months and looked at every cell, at many, many different cells of all of these transgenic lines. So we infected them, and then she looked at 10 hours, 15 hours, 24 hours, and 30 hours after infection and said, does it progress through the infection process like this, or does it get hung up? Does it get blocked? And they broke into three categories. In this case, it looks just like the non-transgenic BY2 cell. So this cell line, this is actually an interesting mutant. This coat protein does not assemble into multimers at all. It's got a mutation in position number 28. And so this guy looks very much like this guy. It progresses through early, mid, and late stages. This guy progresses very much like the wild type, and so forth. This is an interesting group, and it surprised us at first, but not quite. these cell lines are infected with, are transgenic and contain the wild type coat protein. All right, they're more susceptible than these guys. All right, does that go back to what I showed you earlier and said that the coat protein, the wild type coat protein, gives resistance if you challenge the cell with virus. But you can overcome that by challenging with viral RNA. So these guys actually get sicker faster. And there's a whole group of mutants that get sicker faster and more severely than this one, which is even the control. But then look at this group. This group gets trapped, largely trapped, in this phase. They get trapped in making only the small dots or this mid-stage small bodies. It never progresses to large stage, or very few large stage. And this is significantly different from this one based on the numbers of cells identified and so forth. So these mutants are able to give a very high level of resistance, and they're the same group that gave high levels of resistance in the transgenic plants. So then the question was, how do these guys, including this one, which is the one we studied the most, is T42W, how do they interfere with the formation of these bodies? And the only way that could happen, since these bodies are formed by the movement protein, the suggestion was that it has impact on the production of the movement protein and the factories. Right? The virus replication factories make more of these, build on membranes and so forth. I want to talk a little bit about that. So this is what the structure. This is what this infected cell that is of this cell line. Notice its virus replication bodies are largely coat protein and a little bit of movement protein on the periphery and some replicase, compared to the non-transgenic cell line. Very large very large accumulations of movement protein, lots of replicase spots, the red ones, and coat protein is less. So, and these are at 22 hours after infection. So it's pretty clear that the nature of the gene expression in the cell is changed by the presence of this coat protein mutation. If we do this, just section through these by optical sectioning with the confocal and look for the localization of replicase and the movement protein, it was revealed something that Catherine Esau told us years and years ago, that these bodies are complex and replication happens build up on ER. And what we have is the accumulation. You can clearly see that the red is outlining kind of a body. So this is actually the lumen of the ER surrounded by replicase, and outside of that movement protein. There's some association of movement protein with the ER as well. And you can see that even in the very large bodies of very large factories, there's an accumulation that shows the replicase in one spot. This is the tobacco mosaic virus is one of these viruses that doesn't elicit a large silencing effect by the host. It's just not very effective, and doesn't need a silencer because it doesn't silence very much here. And one of the things that we're curious about is how does all this virus replication happen without letting the small RNAs get in? So there must be something architecturally about these bodies that exclude molecules that it doesn't want in. We've done other in situs and shown that these also contain viral RNA, both viral RNA and complementary RNA and so forth. So we learned a lot about the structures from confocal and image reconstructions. We also knew, based upon some imaging, that these bodies really run around a lot. This is an infected cell. This is a section of an infected leaf. You see the stomates. And it's a transgenic line that has the actin filaments lined with talon, I believe. And then it's infected with a virus that makes movement protein with green fluorescent protein on it. And you can see these bodies really do move around. Here's one. You saw this guy come up together and fuse with this one. Here's one that just got absorbed into this larger one. And there's a lot of dynamic movement going on in these things where there's movement protein. We had speculated about 10 years ago that maybe movement protein was one of these fusogenic proteins that just help membranes fuse together. And I think that's probably what it is. It facilitates the production, the setting up of the factory, but it also facilitates the the local movement of these these bodies around the cell. So out of that and a whole lot of other data, we have a model for how coproteins, how these factories are set up and how they might move over to the plasmodesma. Some data that I won't show in this talk really is, I can just summarize verbally, and that is that when the virus comes into the cell, the first thing it does is take its coat off and produce proteins that localize on membranes. The original membranes are around, the first round is on the perinuclear ER. And we were very surprised at first and still don't know how to interpret it. But the red can go onto one set of membranes and green on another. They don't necessarily co-localize. You think about it from a virus perspective, you don't know why it's that way, but it is. We've done immunocapture and have done proteomics on this membrane fraction compared to the fraction that has this, and they are not identical. And then these bodies move out into the cytoplasm. Remember that cell biology cycle I showed you? These guys go out in the cytoplasm, create these large replication complexes. At some level, at some point, the movement protein, with the aid of the movement protein, that these bodies are through myosin linkages to actin, get transported around the cell, moved around. Some of them lodge near plasmodesmata. They set on the plasmodesmata for about 2 hours, and then they go bursting through. So the suggestion is that in that 2 hours, they're modifying in a transient way the structures which allow the bodies to go through. Now, this whole cycle in this cell takes about 18 hours. The cycle to go from cell number 2 to cell number 3 is only 4 hours. So in that first 18 hours, it sets up the factory. And our model is, and the data suggested, including the microscopy, that these factories, as they're set up, and then modify the structure, it's the bodies, it's small factories that go through. They're all primed to get started for the next one. And then they can now establish their factories much faster in this cell than they started in this one, and then transmit it to the next cell. 50 years ago, using fluorescence or polarizing light microscopy to look at infected leaves, virologists were looking at these light reflecting images, which were really inclusion bodies that they were seeing, and they plotted how long it takes for a cell at the end of a leaf. If you inoculated a cell at the base of the leaf, how long would it take for it to go from here out to the end? And they counted the numbers of cells. And they said on average it's about 4 hours per cell. That was done by standard polarizing light microscopy. And interestingly, it's exactly what we found. That once you get established, it's about 3 to 4 hours from here to here. So the old biology works, and this technology told us the same thing, but in a different way. So then we ask ourselves, so how does the movement protein play here, and how might a mutant movement protein stop it from happening? So as I said, we were fortunate to have the structural biology from Gerald Stubbs and other labs. But while I was at Scripps and then at the Danforth Center, we did cryo-electron microscopy to look at virus, mutant and wild-type virus. And I want to point out several things. This is a structure, if you see normal TMV, and if you do a Fourier transform after deflection, you can see that the spacing between molecules is of certain distance, and you can infer from that the nature of the packing. And the wild-type protein would actually stack in a helical way, just as it shows here. Now, just a little sidelight. Stubbs, as he did his work, always found an aberrancy, always found something that not everything was in helical array. He observed this low concentration of another molecule, another aggregated form. And then as we did our work, we found that aggregated form in large amounts was this guy. Now this guy, when it stacks, can stack together, but it doesn't stack in this way. And if you imagine that number A is top, bottom, it's sort of like this. So what's happened is it went like this, that the second group went like this. So instead of A, B, it was A, B, B, A. ABBA, if you like, or not like. But the format of it indicated that might have some activity, and the correlation we have is that when you have more of this, you have less virus replication. In the wild-type infection of the wild-type virus, you get a little bit of this, but we think that what's happened in this mutant is that we pushed this equilibrium much farther in this direction. Then we began to think about that as a regulatory molecule. This guy can actually co-assemble with wild-type proteins. You can get a mixture of them in virus infection. And so the model is that during normal virus replication, you'd have the replicase binding to these regions. And remember, this is the virus I showed you earlier. In the replication cycle, it makes the antisense, and the antisense has promoters that then serve to make these subgenomic RNAs, and these are the messenger RNAs for movement protein or coat protein. And that in the presence of wild-type coat protein, it really turns it on, makes a lot of these guys. I should say it makes a lot of this one, too, to make coat protein, but I don't have more lines down here. So the aggregates are there, and our postulate is that when there is the aggregate of the mutant and wild-type together, it simply reduces the amount of movement protein that's made. So this becomes a negative regulator, probably as the aggregate, because you can't get this if you don't have the ability to aggregate molecules together, coat protein molecules together. So this would act as a negative regulator. What I liked about the model was that, and I think it's right, but we do not have this piece of RNA trapped in that mutant. It means that you can change the function of the protein just as you would change an enzymatic function. You simply make the right mutations and you create the ability to make these aggregates. That's led us into then asking other more difficult questions. For example, when we study, there's another virus that people have used coat protein protection and strategies with, and that's the potiviruses. These are viruses that are sort of in every vegetable and every crop in different amounts, but they're a poti, like potato virus X or potato virus Y or tobacco etch virus. And that one was notorious because the coat protein molecule didn't work. It was the RNA molecule that was the most important. So in that case, you might ask the question, is that coat protein molecule regulatory? And maybe it's not regulatory, but maybe it is. So we did some early experiments and found out, in fact, that we could get coat protein protection as an early stage of coat protein resistance for poties. But what it did was hyperactivate the production of the small RNA. So it's the phase. So if it's not a very good regulator, can you make it a regulator if you know more? And unfortunately, we know so little about the regulation of coat proteins of virus replication by the proteins that are made by the virus. We don't know which one's a positive and negative regulator. Again, the call is we don't know enough basic biology to know which of these will be effective. In the summary so far that we have for the coat proteins, as the mutants that don't form elongated structures don't confer resistance. Mutants that assemble like these restrict infection per se, but not replication. Mutants that have aberrant assembly can restrict infection, disassembly, and reduce replication by reducing the formation of virus replication complexes, thereby enhancing coat protein protection. That's where we stand today. What we've been doing is asking how can we use the science and technology. As George indicated, we have been engaged in work that is relevant in developing countries for a long time, and now I think finally we might actually be relevant, and that's a good thing. And we're relevant, it looks like, in the case of a virus that infects groundnuts, tobacco streak, ILAR virus. And what's fortunate is that ILAR virus is one that was shown by Sulos-Frisch and others early on. It's alfalfa mosaic virus, like virus. And she showed that coat protein is a very good protecting molecule against infection in alfalfa against alfalfa mosaic virus. So that nearly, I guess it was just about 6 months or 8 months after we demonstrated the TMV case. So this is what the disease looks like in groundnut, and it affects 10 million farmers in India. It's really a remarkable disease. It's found throughout the southern part of India, in Andhra Pradesh, and a little bit in other states as well. Some of the early symptoms were confused with others, but it's now known that most of this is caused by infection by this single virus. What makes it intriguing is that the same virus, and we've done a number of, our colleagues in India have done sequencing of a variety of strains, and in fact, the strain differential between these guys is minimal. That whether it's on okra or on marigolds or on cotton or on sunflower, it's the same virus. So that we reckon if we get a good strategy that works in groundnuts, it'll also be useful in these other crops as well. Maheko, a seed company in India, is working on the sunflower case, and others. But we took on groundnut because groundnut is a crop of the poor farmer. It's not sold by commercial seed companies. It's done in the public sector. And if we're successful, we'll help 10 million. It affects more than 10 million farmers. So with a little luck, we could sort of change the production of peanuts in India. So we teamed up with a group at ICRISAT. ICRISAT, because I was on the board of directors there for a number of years and got to know them very well. And we worked together on genome characterization and sequencing and construction. They did the transformation. So the ILR viruses are very different from the tobacco mosaic virus. It has multiple genomes. One of the genomes is cut as the subgenomic into a smaller piece. And these particles are characteristics of this ILR virus. A summary of the results to date. There are lots of transgenic lines that have been generated. And just like coat protein protection in TMV, those that have low amounts of coat protein have delayed symptoms. Those with high levels of coat protein are highly resistant. These are now in the T2, T3 generation, and we're looking for a field trial this year. The Department of Biotechnology in India has taken it up because, unlike the US, it's an interesting system in India. And if you don't mind side comments here. But what we found in India is that the government really cares about agriculture. And they care to the point of providing the science and technology for biosafety as well as the transgenic event. So these guys are getting a little bit of money for field trials for the animal feeding, and they're actually facilitating biotechnology, not standing in the way. And they have a regulatory scheme that's like ours, but it's quite logical. It's not based on process. It's based on what it should be, and that is scientific risk. But we don't know how it works. And the only thing that gives us hope right now is a paper out of Lee Gehrke's lab. For us, we have a model based upon how this virus might work. And Lee's data has shown that there are aggregates between coat protein and viral RNA that are formed. What we found in these transgenic lines was really quite surprising. So one would, and what we know about this virus is that infection requires the coat protein. So why should coat protein protection work in a case like that? What we found, and it's still a little bit surprising, is that when you do analytical analysis of the proteins in the transgenic lines, it's largely as a dimer. It self-aggregates, and so we're wondering now if maybe the dimer is the negative regulator and the monomer is the activator, and that it's that right kind of aggregate which gives a negative regulation to keep things under control. So that's where our thoughts are. Now I want to talk about a very different virus. This is rice tongue row disease. It's caused by two different viruses, one that contains RNA and one that contains DNA, spread by a green leafhopper. And together the symptoms cause the plant to be stunted and chlorotic. This disease is, at least at one time, was ranked by the Rockefeller Foundation as our #1 challenge in rice production. It has since declined in importance because there is genetic resistance to the leafhopper. And largely, it prevents acquisition and transfer, and there's very little resistant germplasm to the virus. We had done coat protein protection assays. This virus is an RNA-containing virus, has 3 different capsid proteins, and we showed that with a capsid 2 plus capsid 3, we could delay symptoms. But it wasn't enough to prevent the disease, because the disease is actually caused by this guy, the DNA virus. And when it's transferred, one can have the disease symptoms form if it's early in infection. Notice that this is a stunted seedling. We all in plant pathology are familiar with the differences in susceptibility of young plants versus juvenile plants versus older plants. So this is the same way here. And we had tried a variety of things. This virus is a pararetrovirus. It has it belongs in this group. It has its own reverse transcriptase, and it makes a single-stranded transcript. On our single RNA transcript that is transferred to the cytoplasm for translation. So during infection, the virus comes into the cell, releases its DNA, DNA goes into the nucleus, forms a mini chromosome, does replication, ships that out in the cytoplasm. The RNA, the transcript length RNA goes out into the cytoplasm as well, and those translation products continue the replication and the products of assembly, and then this is transferred, it accumulates or is transferred to the next cell. So it's a very different virus than the other one, than the single-stranded RNA viruses. It is a para-retro, but there's very little, if any, evidence of integration of this genome into the nuclear genome. There might be fragments, but this is one of the para-retros where there's not a state of integration. We'd studied the genome for a long time. We actually started this project back in 1988, and it wasn't until this year that we actually saw some positive results. I mean, and we had no money for it. After Rockefeller gave up, we sort of put it under the table and would do a little experiment here and managed to get it out. So we're very excited about the results. Otherwise, I wouldn't show the slides. But what we had found out in the interim was that this virus has a very interesting promoter. It's a promoter that works only in the vascular tissue. What makes it interesting for us as well is that this is where the virus is found. Virus replicates and accumulates in vascular tissue. In studying the promoter, we had found that there is a very unique piece of DNA very near the Tata box, and that element is responsible for expression of the promoter in high levels and has some specificity for tissue as well. So we went through the process of identifying transcription factors. We identified two BZIP proteins that form homodimers or heterodimers with each other. They have different structures. We've characterized the domains, each of the domains, and so forth. Then we also, using two hybrid, pulled out another protein that is a negative regulator. So these guys are active. These guys make the promoter work better. This guy slows it down. These can make homo and heterodimers. This guy cannot make a homodimer, but it can heterodimerize with one of these. There's this balance going on. So the question is, what do these proteins do in the plant? And how did the virus come to use them? So this is the promoter. The model is that we know that these guys form homodimers on box 2. The glutamine domain actually interacts with the Tata binding protein in the complex, which then we think activates the holoenzyme to come into this site and then do the transcription run-on out of this major promoter, which is part of the virus replication. So we did the transgenics and said that what happens if you express in transgenic rice the antisense of the first transcription factor, knock it out, and it's pretty ugly looking. But it can outgrow it. All right? This is the antisense of RF2B. And it lets plants happen, but the plants are stunted. And they look a little like tongue row disease. They're chlorotic, they're stunted. This is a transgenic line that has just expressing GUS. And the RLP1 underexpression had no effect. Overexpression weakened it. So again, it's a regulator, but it looks like a different kind of regulator than this. So it became pretty interesting to us that we looked at these factors. And then and it does look a little like this guy. So we did the next set of experiments. We said, well, if that's the case, let's go one step farther. As you know, these BZIP transcription factors have DNA binding domains, and those DNA binding domains will bind this promoter, but also other host promoters, obviously, because this is a host factor. So there must be sites in the chromosomal DNA of the plant to which these guys bind, and they're causing some effects on plant development. So we expressed this guy by itself, just the DNA binding domain. And we expressed it under a constitutive promoter, ubiquitin, and found that it made stunted plants, really stunted plants. We expressed it under the the promoter from the virus and got smaller plants. We expressed it under the CAB promoter, which is expressed the chlorophyll AB binding protein. So it would be expressed in green cells, and there was no effect. We then expressed it under shrunken1 and PAL2, both of which are vascular tissue, and we got smaller plants as well. So the implication was that these transcription factors, this B-zip protein, was binding to elements in the genome which were important for vascular tissue. That would make sense. That's where the promoter is expressed, and that's where the factors are expressed as well. I didn't tell you that or show you that data. And so we ask ourselves the question, what would happen if you put into transgenic plants a construct that just made more of the transcription factor? What would happen? If you overexpress the RF2A or the RF2B and then inoculate it with virus, would it get sicker faster? Or would you make enough transcription factor that the plant would outgrow the virus? And that's the latter seems to be the case, which surprised some of our critics who said that won't work. But it's a reason to keep doing the experiments. And here's the wild type plant that's been. These experiments were done in Phil Rice in the Philippines. They challenged the plants with insects that were carrying the virus. So it was a greenhouse experiment. This is the parent line, and you see that it's smaller, it's shorter. This is their local susceptible control. And you see that it's like our control. This is a line that is resistant. It's their normal resistant cultivar, the one that's most resistant to insect feeding. And it gets pretty well grown up. It seems to be growing out of it. These are transgenic plant lines that overexpress the RF2A, these two lines 4 and 5, or RF2B in 6 and 7. So it looked pretty good that they were able to withstand the infection and sort of live through it. And this is, again, a reiteration of overexpression of. So we now looked at the impact of elevated levels of these two proteins on Tungo disease symptom development. And I should say that, yes, these do cause the expression and the accumulation of more RF2A. It's not an antisense, but you can measure more RF2A and more RF2B in these lines. And this is what the data showed after. So each of these lines represent measurements of the height of 30 plants in the pool in our growth chambers in St. Louis. Now, by this time, we developed an agro-inoculation procedure, so we could inoculate by using agrobacterium to deliver the virus. So if we do that on the control line, you can see that 3 days, within about 35 to 40 days after inoculation, the plant stopped and they didn't grow anymore. So that's about what we expect to find. Yes, please.
Do you have the accumulating virus? Is it virus tolerance or is it suppressing viral development? Sorry.
Time's not up yet.
Yes, sir.
I get to tell you the answer. And these are transgenic lines that contain RF2A. They continue to grow. And these are the lines that have RF2B. Notice the profiles are different. There's a little bit more elongation towards the panicle formation in these guys. And this is a non-transgenic control. So they grew equally as well or a little bit better than the control. And the yield of the seeds was about the same. So it seems like it outgrew it. So then and this is the plot that comes from the lines done in Philippines. This is the resistant line by their native resistance. And you can see that the lines that contain RF2A are as equally as resistant as their best insect-resistant line. So here's the experiment. So we then made protoplasts out of those rice lines, out of all 4 of the rice lines, and made protoplasts so we could study single-cycle virus replication. What happens when you inoculate these cells? Take away the leaf, take away the vascular tissue, take away it all, and are these resistant or susceptible? And this, I didn't expect this. I thought the resistance would be effective in the leaves, but not in the cells. In fact, I asked the postdoc not to do the experiment, just get on with writing the darn paper. And he did the experiment, and it's led us into a whole new area. And I won't show you all the models, but so here's the amount of accumulation of viral RNA of transcript in these cell lines, and you can clearly see there's less transcript in these cells than in the cell line of the non-transgenic. Similar things were done with sets of infected plants. This is a quantitative RT-PCR. And it's the accumulation of viral RNA in virus in agro-inoculated plants. Again, 30 plants in a population, all mixed together and averaged out, right? So this is viral RNA accumulation in the control. It peaks at about 21 to 24 days and then lays down to a level and continues on forever. And here, then this line, you can clearly see that there's less viral RNA made in this line that has more RF2B. RF2A is also a suppressed one. Then you did virus accumulation, and the same thing happened. So these plants make less transcript than the controls, and as a consequence, less virus. Now, it's not immunity, but if I was a betting person, I would say if you had that level of resistance, you wouldn't have epidemia because you couldn't have transmission to the next line. So that's what we in plant pathology are all about, reducing epidemia. And so there's a hope there that this might actually be useful. And so the model is that as the virus is delivered into these plants, it uses the RF2A and RF2B and all the other regulators and causes a redistribution of those factors to make virus instead of making plant growth. And that what we've done is overexpress these limiting factors and now are able to do this. Now, it's also possible, as it reorders gene expression to get stunting, the other question is, what else does it induce? And very interestingly, Shanghong Dai, who's doing this work, went ahead then and took these plant lines and then went through a bunch of pathogenesis pathways and said, what does it do to the MAP kinase pathway, to the PR pathways, and so forth? And to my absolute surprise, it changed a number of them. Not all of them, but a number of them. So it's not just apparently. I was naively thinking the simplest explanation is the plant just grows through the cycle, this phase, very quickly because it's got enough transcription factor, it just gets through there. I think that's probably right. But I think it's also maybe affecting other pathways that affect innate host resistance. And those together are what suppress the amount of replication. So it's open for us a new field, and this is a project that Shanghong Dai will take off with him when he goes into his own independent career. But it's very exciting, and again, it's because he did something I said don't bother with. The other approach we've taken is, sorry, let me back up. The cool part about this is that there are many different strains of rice tungro virus. The strains in South Asia and India are different than they are in Southeast Asia. So I'll be going to Delhi in a couple of weeks to see, to make experiments there, get experiments started there to test it against the Indian strains. And my guess will be that it'll be effective against maybe a pan resistance across many cultivars, and strains rather. We've also made synthetic zinc fingers to bind to specific areas. These plants are now in the homozygous state and will be challenged later this summer. But I can tell you that we've done this with synthetic zinc fingers. Some work better than others. Here's a zinc finger that shuts down that promoter in transgenic Arabidopsis by 80%. And we have that now in transgenic rice, and we'll be testing it out. So we're optimistic that we may have some effect by a totally separate mechanism using synthetic zinc fingers. The last example of protein-mediated resistance I want to talk about is in the Gemini viruses, and I'll just show a couple of slides from Claude Fouquet. Claude's lab has been studying these Gemini viruses in an African host, cassava, and the challenge will be to get this out into the field. Now, this is the disease as it looks. The virus is a geminate, and it's a bigomo or virus, and it has two different strands of RNA, of DNA, single-stranded DNA. He's taking a hairpin structure approach for silencing and has some transgenic lines. And he's also taking the G5 or so-called G5 single-stranded DNA binding protein from an inovirus. And that sequence, it's been modified with new codons and so forth. And we now have a number of lines that have resistance to this disease, to cassava mosaic disease. What we like about this one, unlike the single-stranded RNA approach, this protein gives resistance against the Sri Lankan strain, which is very different than the Uganda strain, and it gives equal resistance against both. So it looks a little bit like this approach, a protein-based approach, will be effective against broader ranges of Geminis, not as an immune reaction, but as high level of resistance. So we're optimistic that strategies like this, which slow down disease progression and virus accumulation, will be effective under field conditions. So this project is now transitioned from research into what we call pre-commercialization. And now you're going to hear me talk like a company guy. Because as an academic, we don't have a clue about how to get a product developed. So we submitted the sequence and the protein to a consultant that tells us if it's likely to be a problem with an allergenic in the food. Then we went on with creating processes, all the way from barcoding to lots of processes to try to get these products into a situation that we could submit them for field trial in Uganda and Kenya. Two problems here. Uganda and Kenya don't yet have a biosafety protocol for commercialization. Fortunately, they have in the last 24 months established a protocol for greenhouse trials and confined field trials. And we've worked 4 years with the Ugandans and the Kenyans on that protocol. We did it ourselves. We got involved with some resources from USAID and helped to teach, and we will benefit from it. So these are under review by the institutional and then the national biosafety committees. And we expect to be in a confined field trial in Uganda in the April, May, June timeframe. In Kenya, we're not sure. We were set back by the civil disruption there earlier this year. It was in the Rift Valley that we wanted to have these trials. So we've been set back at least 5 months in Uganda caused by that disruption. More importantly, we're now doing this in farmer-preferred cultivars. What we've done is a model plant. Nigel Taylor, who is the tissue culture guru for cassava, is now showing that he has transformation in at least one or perhaps two cultivars. So this is the work of Claude Fouquet and Nigel Taylor, and he has a wonderful group. But what's made this work for us is that we have at least 4 from the private sector to help us know how to do this process. And that's because of this. To develop a transgenic a line of transgenic plants, get it tested, get it to the field and to the farmer is a long process, and you need enough funding to get from here to there. But it's not simple. If it had not been for information shared by our friends across the street from the Danforth Center, people at the Monsanto company, if they had not shared with us their process, I don't know that we'd be as far along as we were. Monsanto's been a great partner in this whole process. They have nothing to gain from it. This is a strict giveaway, philanthropic kind of thing. For us, there have been no patents filed on any of the technology. The material will be given away freely to the national program staff, and they will take it from there. But this project really started in 1988. We had the first resistant plants in 1999 rather, 11 or 12 years after starting. We were all excited about the first line. After 3 years, it went silent. The gene silenced. We went back and looked at the genome structure. We should have known better. It had 7 fragments in it. We should have known more. But we knew it had fragments and had gone back already to start over. And so the reiteration, we've repeated it and now added the G5 construct to the process. And we're here, and we're beginning initial confined field trials in this year. This process will take different times for different projects. I mean, one expects if you had a BT project, it would go a lot faster. But when you're starting with developing the technology, the knowledge base that would actually make a product, you really have to think differently than when you're just doing research. And we've learned that in spades in this cassava project. It now teaches us about, we have a maize project and a soy project and a sweet potato project that, if we're successful, will lead to new products as well. So it's informed everybody in the Institute that wants to do biotechnology about what the challenges are in getting through this process. So do I think it really will take 10 plus 6 plus 5 plus 3 plus 10? No. I think from the research side, it took at least 12 years here, and it's getting, that time frame is about right. This time frame ought to be shorter if the trade is good. If it's not, if it's a failure, we've got to go back here again. But it's lengthy. And the question for academic institutions is, how in the world do you get the money to do that? And since it's done by graduate students and postdocs, how do they publish out of it? Because it takes 8 years to find out if you're successful. So you have to realize that this is a different workforce than the workforce that does this part, and gear up for it. Maybe that's why there's only one product from the public sector that's in commercialization. Remember my second slide about it's only papaya so far. There's probably a reason for that. And the Indians seem to have gotten it right. That is, the government's going to get involved in the process of research and biotechnology and commercialization. And that's a different paradigm. We don't have that in this country. So maybe it's the states that should get involved, or maybe it's the grower groups. It's not going to be the federal government, I don't think. But how we play in the field of advanced technologies, including biotechnology, to do our jobs as agriculturists, I think still is an open question in this country. So that's what I've talked about today. I'm a protein kind of person. If somebody discovers some RNA, I say, give it away or publish it and move on. But I really think that the protein side of things has been, for me, the most exciting. I can see structure, I can see accumulation, I can really sense how things work. The people who did the RTBV project, Shanong Dai has been the leader in this area. We've had help throughout by research assistants in here. Carlos Barbas at Scripps was a collaborator on the zinc fingers, and Antonio Alfonso is the Phil Rice collaborator in field testing there. In the case of TMV, this group has now largely dispersed. We do very little with TMV except on the cell biology of infection. I lost my NIH grants and didn't, I think the duties in the office that I sit in is a little different. And I fault myself for not getting more publications out of that, and it's my own fault. But there are other things I have to do besides just think about science, and I love coming today to talk to you all about what you're doing. It gives me more hope that my mind can still work and I can still grasp things. Thank you very much for your attendance and for your participation.
We have just a few minutes before Roger's next appointment.
But I'd sooner answer questions. Yes.
A technical question. In the case of the rice pararetrovirus, you were using as a control that variety which has resistance to the vector.
Yes.
But I was confused because I thought at one point you indicated that you were using agroinfection to deliver your virus.
In the case of in the case where we use the natural resistance, that was done by insect transmission in the Philippines.
Okay, so the comparison there was across.
That's right, and we have not gotten MTA to bring their rice varieties for us to check with agro inoculation.
I see.
Good catch. Yes.
When you were looking at the rice tundra virus and the RF2A and RF2B, did you ever put them together in the same plant?
No, the crosses are being done now. We had the homozygotes at least a year ago, but we have not done the crosses and get them both in. What we're seeing is that there are some very interesting phenotypes in early plant development, and what we're hoping is, of course, that there'll be something to really stimulate higher levels. But there's lots of stuff we don't know about the factors. I mean, we know that virus infection does not in itself stimulate the production of RF2A and RF2B in the non-transgenic lines. It doesn't induce the production of more. We don't know we have a process of trying to identify the host genes that are controlled by RF2A and RF2B using a genetic approach, and have several that look quite interesting. But that's right. Yeah, that's an interesting set of genes downstream of those factors, but there's much to be learned about what might be causing resistance. So all that Shanong has done so far, what he's done so far, is to measure the accumulation of certain RNAs of other disease defense. We don't know what they do.
Other questions for Roger?
Yes.
What are the thoughts around virus overcoming the resistance brought up by transgenic strategy?
The thoughts about virus overcoming resistance caused with transgenic strategies. In the case of a coat protein, I'm reasonably optimistic that it'll be relatively durable because you'd have to basically change the nature of replication and regulation to overcome some of these things. In the case of the single-stranded DNA binding protein, that seems to be effective against all Geminis so far tested. Not immunity, but a level of resistance. So I think that's going to be a tough one. I think that might be a durable one. In the case of the transcription factor, we don't know. I hope to find out by the end of this year if we can get it challenged by Indian virus. And the Indian strains are about 15 to 20% nucleotide divergence from the Southeast Asian strains. So if it works the same way there, we would be really pleased, of course. And I think that's that's really the reason why I favor protein-mediated events. Once you know what mechanisms there are, you can act like a biochemist and make the changes that make them better at what they're designed to do or change a bit of the structure. It's not to say that the small RNAs won't be equally as effective in some cases, but in the case of, for example, there was a paper published just very recently in India, a short RNA against rice-hunger disease, and I think it was in Transgenic Research last month. And in fact, they got good resistance, not immunity, but reasonably good resistance against that virus. But my guess is it would not be resistant against the Southeast Asia strain. So I think it probably depends upon, the choice will be depending upon the virus and the nature of the diversity of sequence in the population. Virologists have known for a long time that plants are infected by swarms of virus, not one strain, but a whole range of sequences that are some of which are successful, some of which are not, and that where there's more divergence, I think the challenges will be greater.
The whole durability question has to be assisted by your discovery of distinct mechanisms for protection, because a virus trying to overcome two different mechanisms at the same time is going to have a very tough time.
It'll be a tough time. Yeah, that's right. Thank you all for coming out, and for the time that many of the students and postdocs and faculty gave today. Thank you very much for your interaction. I enjoyed every bit of it. Thank you. You're very welcome. It's a pleasure to be here.